Last week, we have explored the foundation nexus of RAS fish production, hydroponics and bioponics, and microalgae integration. We have been turning waste into wealth, and we use Ampli Aqua as our baseline. And today we will dive a bit deeper. We will go from wastewater and effluent to soil fertilizers, microalgae, and biostimulants. We will kick off with Aqua Microbiome Database, which is our real-time tool for decoding water microbiomes. We will have Jorge Antunes showing us how to monitor microbial dynamics in aquaculture, wastewater, and how to unlock valuable nutrients, turning waste streams into high-value products such as biostimulants and biofertilizers. Then we will have Jon Brage, which will demonstrate how concentrated RAS effluents can replace mineral fertilizers for plant cultivation, creating this time a closed-loop system that eliminates chemical inputs. And later on in the session, Etiele Morais will close with wastewater for sustainable biofertilizers and, of course, biostimulants, this time from microalgae, showing us how we go from lab validation to industrial scaling up. I think this is going to be a promising session, and also these insights connect directly to our work here at Food4Sustainability, where we bridge wastewater, microalgae, and soil regeneration. And as you can see in the slide, the Regenera Lab, which is our soil health and function laboratory, will provide microbiome analytics and practical decision frameworks for transitioning to regenerative soils. Also, Ampli Aqua, that you should know by now, generates the concentrated RAS effluents into this nutrient-rich stream, aquaponics and bioponics, and onshore MTA systems, as we have seen during the last session. In the meanwhile, it's in the field through Idenha Rural Food Park that it serves as our rural innovation hub, where we link biostimulant production, processing pilots, and farmer entrepreneurship. And this all happens in Idenha region, as you know, it's in the interior of Portugal. So, turning now to our experts for today's session, I have the pleasure to introduce to you Jorge Antunes, is the microbiology specialist at Food4Sustainability. George holds a PhD in biology, specializing in microbial communities and bioactive properties. George's environmental research and scientific publishing experience deliver cutting-edge insights for sustainable food. We also have the pleasure of being joined by Jon Brage. He's a scientist at SINTEF Research and also an associated professor at UIT. Jon leads SINTEF's Circular Value Chains Group, focusing on aquaculture sustainability, and his marine biotechnology PhD bridges research and industry for circular economy solutions. And also, Etiele Morais, she's a researcher at UPC Ramon and Cajal in Spain. And Etiele specializes in microalgae cultivation, wastewater nutrient recovery, and biostimulants, biofertilizers. And from GEMA Group, Etienne scales solution from the lab to the industry. Thank you so much for being with us today. And just before I turn over to our dear speakers, let me just remind you some of the house rules for our webinars. As usual, we are going to record this webinar. if you're not comfortable with the recording, I will need to ask you to switch your cameras off. We also welcome your questions. We would like is we all learn from your questions. So we will have questions at the end of each presentation. Let me remind you that the questions will be done through the chat, either here in Teams or the chat in Slido. And we will have a look at them and direct them to our speakers. And also at the end of the session, we will have this very short, simple quiz. This is important if you want to get your certificate. Please don't leave without filling in this short questionnaire, okay? Thank you so much. House rules are established. And now we need to warm up into our topic. That's how we always start our sessions. And let's see what the team has prepared for us today. You know that you can scan the QR code to join Slido, or alternatively, you can use a browser in your computer, in your PC. It's very easy. Just search for www.slido.com. In the hashtag box, please just insert our code. It's always the same, F4S Academy, and we should be ready to test our knowledge. Okay, so I invite everyone to jump on Slido. This is more fun when we are all trying to learn at the same time, also try to learn from each other. So let's see how many of you have already joined. Our speakers are also invited to join Slido. Okay, here we are. So the first question is, what dual role do microalgae pull off in sustainable farming? So what dual role, two characteristics that microalgae can achieve towards sustainable farming? Is it about the carbon sink,
Claudia.
pollinator being a pollinator booster?
Claudia,
Yes.
sorry. Can you please try to switch off your camera?
Okay.
Because we
That's
are.
the internet's.
Yeah. Thank you.
Okay. No worries. I'll do this. I hope it has improved.
Yes, a little.
Hopefully.
Mm-hmm.
Okay.
Yes, you can go.
Yes, thank you. Thank you, Soraya, for letting me know. So the questions are carbon sink and pollinator boost, biofertilizer and biostimulant activity. They can be a mycorrhizal enhancer and salinity reducer, or microalgae can be both pest repellent and a drought shield. Let's vote. And I will start to go through the questions. So a carbon sink and a pollinator boost. Microalgae, as we know, they sequester carbon, but they don't directly boost pollinators. Are they mycorrhizal enhancer and salinity reducer? We know that microalgae influence soil microbes indirectly, but they are not a primary mycorrhizal partner. What about being pest repellent and drought shields? What we know is that we have some promising studies at the research level, but we are not have conclusive studies in terms of field about these capacities from microalgae. So what we have here is that the real winner, as you correctly point, is that they can be both biofertilizer and biostimulants. Let's just understand a little bit why, because microalgae deliver essential nutrients, namely nitrogen and phosphorus, and also some micronutrients that can be used as biofertilizer, while at the same time releasing phytohormones, amino acids, and antioxidants as biostimulants. And this assists in stronger roots and stress-proof crops. So well done, everyone. So we already know something. Biostimulants and biofertilizers can be enhanced by microalgae. Let's see if we have that's our second question. This now points us to what parameters should we control. So which parameter is critical to control during microalgae cultivation, because this parameter affects both carbon availability and nutrient solubility. Okay, which nutrient? Which critical parameter? Apologies. Turbidity, pH, dissolved oxygen, salinity. So when we are cultivating microalgae, all of these four parameters are important. Which one is the most important? Would it be turbidity? As we know, turbidity mainly impacts light penetration for photosynthesis. Dissolved oxygen, it's also important. It affects respiration, but doesn't control carbon speciation or nutrient precipitation. Would it be salinity? We know that salinity influences osmotic stress, but does not dictate nutrient solubility chemistry. So here again, well done. pH is the critical parameter to control because it determines the carbon form, whether it is CO2 or bicarbonate or just simply carbonate for photosynthesis, while at the same time preventing nutrient precipitation, such as phosphorus, iron, and trace metals. So a practical takeaway to bear in mind is that pH control is equal to carbon availability control in our bioreactors. And hopefully we'll learn more about these processes throughout our session. Well done, guys. I can see a big green tab in the answer, so well done. Let's see if the third question is a little bit more challenging to you. Slurry. Slurry that comes from harvesting algal biomass is what we do with the slurry that is obtained from harvesting algae. It's anaerobically digested for biogas, is used as a biofertilizer, is discarded, or is it recirculated? Okay, so anaerobic digested for biogas, used as a biofertilizer, discarded, or recirculated. Okay, I see more answers, so more variability in your answers, which is good. So we need a little bit of explanation. So anaerobic digestion for biogas. This is not the correct answer because anaerobic digestion produces biogas. It's true, but the digested slurry becomes the fertilizer. Okay, so this is not. Do we discard this slurry? We shouldn't be discarding this slurry because we would be wasting valuable nutrients. So discarded is not the correct one. Is it recirculated? Not really, because recirculation keeps the nutrient recovery value. That's what we are actually looking for. So the correct answer is slurry obtained from harvesting algae biomass is used as a biofertilizer. So after microalgae clean the wastewater, their harvest slurry delivers slow release NPK and also micronutrients, which improves soil structure, boosts microbial activity, and close the circular loop. So this is how aquaculture wastes are really turned into regenerative soil power. So well done, everyone. Thank you so much. I think we now feel a little bit more warm. So it's now time to turn to our speakers. Let's get ready for these actionable insights of closing these loops from aquaculture waste to regenerative soil health. So Over now to our speakers. I will now call to the floor Jorge. Jorge is going to be the first one, first speaker. So whenever you're ready, the floor is yours.
Thanks, Claudia. Please let me know if you can see my screen already.
Yes,
Yeah,
all
we're
good.
in
Perfect.
presentation.
Is already presentation mode, I believe.
Okay.
Yes, all good.
Thank you both. Thank you, Claudia, for the kind presentation. And good afternoon, everyone. So my presentation is entitled Closing Nutrient Loops and Integrating Microalgae with RAS and Agriculture for Sustainable Food Production. First of all, I would like to raise your attention to the issue of increasing nutrients demands. So currently, in terms of the consumption, we can see there is a increasing consumption of nutrients worldwide that is related to the industrialization and developing countries. As you can see, most of the developed countries already have a high share of adequate nutrients in terms of their consumption. However, the developing countries in the near future will also increase their share of those nutrients. In terms of food groups, I would like to highlight that proteins specifically will see a very high rise of consumptions until 2050 if the trend continue. As you can see, we can expect a yearly increase of around 3%, which is quite significant. This, of course, will result in really high demands in agriculture and aquaculture production. That this dietary shift towards more rich protein foods will encompass. This, of course, will have to have a response in terms of production. Now I'd like to raise your attention to the production of agriculture. You can see here the increasingly rise of fertilizers production and respective markets. So to give an answer to the rise of consumption, there is a high production of fertilizer to increase the productivity of agriculture. Same way, the aquaculture, which is the most significant source of protein in the future, is also having a really significant increase, as you can see, until 2020, and the trend will continue. And also, I'd like to highlight to you the aquaculture production is mostly increasing in inland water intensive systems. Overall, the agriculture will need much more production, and this will mean higher fertilizer production. The aquaculture as well and respective operations will also need a much higher fertilizer production, mostly nitrogen and phosphorus inputs. However, this is a problem because currently there is a very high nutrient losses in food systems. So traditionally in agriculture, with the fertilization of the soil and with the runoff with water and rainfall, a good share of nutrients will go and will be leached into the soil, and this will go to be incorporated into receiving water, and a good amount of these nutrients will be lost. This is, of course, is a problem, will lead to water pollution and a significant loss in resource efficiency. The same panorama we can see in aquaculture, mostly in intensive open aquaculture, where a large fraction of nutrients, again, mostly nitrogen and phosphorus, is not retained in the fish biomass. In fact, it's even lost in the water, wastewater, and this is also a problem in terms of pollution, but at the same time can create some recovery opportunities. A bit similar with the agriculture perspective. Aquaculture can also, the loss of nutrients can also impact fish health in this case and system stability. Now I would like to show you some interesting data. So just really to quantify these losses, you can see some data regarding aquaculture and farming production in the UK. You can see that in the first two lines there is a good amount of nitrogen and phosphorus that is not accounted, so it's lost. Interesting, sometimes it's not really know where the share of these nutrients are lost. In the aquaculture, and this is referring to open systems, a very important amount of nutrients, so up to 85% of feed nitrogen and phosphorus is also lost to water or to the sediments in the respective environments. To deal with this nutrient loss that has been faced really since the beginning of industrialization, there is new concepts that can deal with this. So starting with agriculture, there is the concept of circular agriculture, which is essentially a transformative approach that can focus on a more efficient use of these resources. So try to use some of the effluents and some of the wastes and put back in the soil and make the soil much productive. So closing these nutrient loops. And it's a very important concept that has much more interest nowadays. As a whole, this should have a higher recovered nutrients, higher crop production, as well higher nutrient recycling in soil, and overall a better soil health. Regarding aquaculture, I also like to introduce you the concept of recirculating aquaculture systems to the ones which are not familiar with this. So this is basically aquaculture system in which a very high share of the water can be used. In this diagram, for instance, up to until 98% of the water is reused. This is also a system that is with a control environment and with improved feed efficiency that will result in less feed to the fishes. There is so less nutrients used, and as an output, there is also a higher concentration of nutrients such as ammonia, nitrate, and phosphorus, which can be a problem, but can also be an opportunity. Now I would like to introduce you to the possibility of using microalgae as a biological nutrient recovery tool. So I introduced the concept of RAS and also circular agriculture. However, this system in itself, even though they are a major improvement, they are not perfect. And here microalgae can help to make this system more circular and much more efficient. Here I think we have some interesting data regarding microalgae. As you can see, some species that are represented here can have a very high removal rate of nitrogen and phosphorus removal and can be used for different systems according to their efficiency. This will make their use very useful both in agriculture and aquaculture. In terms of biomass production, and here we have a raceway system, which is open system production in our partner facilities of Ampliaqua. Microalgae are a very important biomass production of different nutrients. We can have from proteins, lipids, vitamins, and pigments that can be harvested and used for different goals. And again, depending on the specific species and also in terms of production, they can be optimized for different goals and be used for different valorization. The most direct and most obvious use of this microalgae is a biofertilizer in agriculture. They can be used directly in the soil. They can be used in the leaves. You can see a very interesting diagram here in everything that they can be involved in. Just giving you examples that without going into a lot of detail, that several microalgae species can have a major improvement in the health and production of different crops with very economic importance overall. Likewise, in terms of aquaculture, microalgae can be used in different ways. They can be dry, they can be used as a paste and pellets, and they can be used for biomass production. They can be used as part of the feed, but also very importantly, they can be used to help improve the health of the fish, and this will help also replacing other traditional fertilizers that have higher environmental impact. Now, this will relate, how can we relate microalgae and aquaculture, specifically with RAS recirculating aquaculture system? Without going into much detail, microalgae can use some of the wastewater from these systems, like I showed you before. They can uptake nitrogen and phosphorus. Some of this biomass can be enriched and can even treat some of this wastewater, which again can be used and be inserted again in those systems and be part of a very efficient circular system. Just to give a final panorama of this opportunity of the microalgae in aquaculture. So the effluents of wastewater from RAS, which have high nitrogen, phosphorus, organic load, and solids. With the integration of microalgae, there is high benefits since they can be used for the removal of nitrogen, the phosphorus as well, reduce the share of organic pollutants and be used to produce biomass and recover some of these resources. However, given this panorama of the RAS, which are absolutely a very important tool for the circular use of nutrients for the future, with much higher use as well in the future. There are also some problems that affect these installations. Starting with the system complexity, overall RAS are systems which have some complexity in terms of process, interactions, systems, compartments that make this their monitorization operation a bit more difficult. These systems, since they are intense, they can build up nitrogen and phosphorus concentrations, which can also be a problem when the treatment of wastewater is the most correct. Since it is also an intensive system, this can cause risks in terms of microbiology. So some pathogens like bacteria and virus can be more common and create disease outbreaks. At the same time, there is also the difficulty of monitoring all these difficult, complex systems in real time. We do have this challenge, but we fortunately are also in the point there are very important innovations that can be applied to RAS, and I believe we are in the right path for this. So using sensors, digital platforms, and IoT and microbiome monitoring, such as NGS platforms that have seen a real good development in the last years and have become much more accessible and cheaper. We can use these technologies to make the RAS systems more efficient and the management of their nutrients and waste much more efficient. And given this long introduction, in fact, I would like to pass to the project that we are developing here in Food4Sustainability and also with some partners, namely BGI here also in Portugal, and also with some German and Norwegian partners as part of Eureka project. So the project is called Aqua Microbiome Database, and it's focused on creating technologies that can help monitoring and optimizing the RAS systems. This will act in the intersection of the environmental problems that I was talking to you about, and also about the fish diseases and the pathogens that I also explained to you are a big problem in these intensive systems, because very much since a pathogen is present, given the high concentration of nutrients, sometimes the diseases can show up really quick, and it will be very difficult with the current technology to control these diseases. So we try to address this solution, namely the appearance of these diseases in fishes in the systems. And here you can see our RAS systems in Apliaco where our project will be implemented. We are considering developing different tools. Not consider, actually, we already developed these tools. We are developing, with the help of our German partners, two types of sensors. These sensors, without going into much detail, will allow to detect minor fluctuation of bacteria pathogen presence through their metabolism, essentially. This will allow to give some red lights to know that the presence of those pathogens are there, almost in a real-time way, which will allow to detect these pathogens really quick. At the same time, we'll have a water microbiome analysis platform help interpret these results and also to characterize these communities in the aquaculture systems, and at the same time a database incorporating information about these pathogens, their meaning, how to deal with them, and also how the pathogens are related to specific nutrient concentration will also be built. So the database will incorporate reference to reference papers, to also to reports, to give highlights of what could be happening in the aquaculture systems. With these solutions, we believe that we have the right tools to be much more preventive and to help producers of aquaculture to act before time and avoid the appearance of the diseases. So we'll have tools to monitor water in real time. We can do assessment of microbiological safety. At the same time, the wastewater from these systems, as I explained to you before, can be much more monitored. So we'll know what is the quality of the wastewater when we will discharge them. And our database will allow to have give the producers much better management decisions, improve the fish production and the sustainability of the soil systems. This is an ongoing task at Food4Sustainability, and we believe we'll have an operational version really soon that will be optimized further, but can really soon give some advices to producers to know what they can do when they have a pathogen presence in their aquaculture. Overall, this project will help make efficient RAS systems much better. Main highlights: improve water quality, better productivity of fishes, especially fishes with economic importance, to help replace, give protein and higher food production, decrease disease outbreaks, and also have a better nutrient management in the systems overall. Really, to conclude and to make the final picture to you. As I said, we have a major problem of nutrient loss in agriculture and also aquaculture. However, we are with the technological innovations we're seeing currently, namely the appearance of circular systems, biological recovery brought by microalgae and other organisms, and innovation at aquaculture systems, we can make resource recovery much better, really reduce the amount of pollutions that is being liberated by these systems, and just making the food production much higher. So will really be crucial to make the production of food that I said in the beginning will really be necessary in terms of outputs, because the population is increasing and is demanding much more food production. However, we really need this efficient system to make this production efficient, sustainable, and transform this nutrient waste into value production as well. Just to conclude, I'll also leave my contacts and the contacts of my colleagues in my projects. If you have any questions about this project and specifically the database, I'll be happy to answer your questions right away. Thank you.
George, it was very, very interesting. So to kick off discussion, I'm going to start with a question about the aqua microbiome database. So, George, you clearly identify that we have here a technology platform. This allows real-time monitoring and the optimization of RAS systems. My question is, could this at some point be transferable to other wastewater streams, like, for example, industrial ones, like, big producing juice companies, where we know that there are a lot of nutrients that are not recovered through their wastewaters? Or do you see aqua microbiome database only as a RAS system?
Yeah, very good question. Well, I think the benefits of this project, although we didn't see it during the proposal and the writing of proposal, it is really scalable. So the inputs and the gathering information that we'll collect can, I believe, can be really transferable to the other system, because it's the really good thing about this process is the synergy between sensor technology, the gathering of data, and also biotechnology data and informatics that can create that database that you mentioned. Then, if you want to escalate and transfer to different kind of production systems like food production, or like you said, it will be essentially a matter of updating the data reference to know what are the specific diseases or contamination. I believe, for instance, if you're talking about fruit, probably the most concerning microorganisms and diseases could be fungi. So it'll be a question of updating the databases, know what are the real problems, and also to have in mind what will the end user want to know. For our current database, we know that for fish producers, it's important to have integration of data about fish health, about the characteristics of the aquaculture system that I talked to you about, but also information about the symptoms and the pathology of the fish. This needs to be really, really have a good synergy, and that's what they've been working on because the producer wants to know what are the red lights. So we have this symptom in the fish. I'll use our database to let me know what can be happening real time with our fish and with our culture. If we transfer this system to other kind of productions, we also need to know what are the major concerns, but the platform that we already have, for sure, I think it's really scalable for that.
Thank you, George. Very, very interesting. I think we are really walking steps towards a real circular world. So that's good to see. Good, best of luck with the aqua microbiome database. Johan, is nice to see you again. Thank you so much for your question. I was going to ask your question to George, but unfortunately your question has disappeared, at least from my screen. You had a question about uvas, which is the sea lectures, and you were referring that you use them at your MTI systems. Apologies, something went wrong here from our end. So if you would be too kind just to post again your question, we would truly appreciate, and I will direct it to George. George, in the meanwhile, we have another question here. So what are the main obstacles for us? Why isn't more
Claudia, we are experiencing
yes, everybody would like
some
to hear.
I lost you there, Claudia.
Sorry for this. So I was just saying that we want to know why, what are the main obstacles for us, and why isn't more dissemination in the aquaculture sector? George, what are your views on this?
Again, good question. I believe it's mostly so far a question of investment, so basically capex. So it's relatively expensive compared to other aquaculture systems. So traditional aquaculture system, you could implement them almost in short. So open system that water would just get in and get out, and you can add freely the nutrients. This has systems that I'm trying to explain a bit in my presentation are definitely complex. They need to have different compartments where the water recirculates, needed purification, sterilization of their compartments as well. They need, of course, also experts, technicians dealing with that to build up the system and also to operate this. So I'll say the most difficult aspect is investment, I think. Otherwise, yeah, and also the challenge that I'll also try to explain to you about operation. Sometimes producers can already have those systems in place, and they can have the funding to have interesting financial outputs of producing certain species with those systems. But sometimes there are pathogen outbreaks or fluctuations of temperature that these systems are much still much better than traditional ones, but there are still some challenges that can raise some difficulties. But these issues are also really being answered to with those technologies that I mentioned. So the sensors and also a better understanding what's happening in terms of water quality and microbial quality and fish health will make these producers much more happy with this. So I believe, at least in developed countries, this will be the most likely the most used production of aquaculture in the next decades, for sure.
Thank you, George. I would also add that at least now we have this community here, so we can start spreading the word of aquaculture and its benefits and how it can play an important role in the environment. And it will start as a good starting point to overcome some of the challenges that George was just mentioning. Very, very quickly, George, because I know that you're going to give a quick answer to this one. So will Aqua Microbiome Database develop the prototypes for the sensors? Very quickly, because we are short on time, George.
Well, we will have a prototype for the database that we will have soon, and that we can share with some producers of our network, so to let us know if they're having the information they want. In terms of the sensors, our colleagues are working on that independently and also to optimizing. But in the future, yeah, basically the prototype of the sensors will be integrated with the database, and users can see if there is alteration on, let's say, pH or something that related to metabolism of bacteria using those sensors, they can use already the prototype of our database to let us know what is happening in terms of microbiome. So we'll hope to have those prototypes soon.
Thank you so much, George. Very, very
Thanks,
good.
Claudia.
Let's definitely be in touch to the developments of Aqua Microbiome Database. Thank you so much, but we need to move on, and we already have a line, John, to jump to the screen. So, John, whenever you're ready, you can start sharing your screen. Thank you.
Thank you. If I can find it.
It was there. We have seen it before.
Yes.
It's there. Not in presentation mode. Now in presentation mode. Thank you very much. You can go.
All right. Well, thank you for the introduction. And I'm going to talk about how we can recover some of these nutrients from raw effluent. So I think it's a good overlap between me and Jorge from the previous presentation. Anyway, my name is Jon Brage Svenning. I have a PhD from UIT, which is the university of my hometown. I did my PhD on lipid chemistry in these mass-cultivated marine diatoms. And I've been employed as a research scientist at SINTEF since 2022. And SINTEF, for those of you who don't know, is a large research institute here in Norway. We have about 2,200 employees. And sort of our vision is to create technology for a better society. So our whole business model is to work closely with private industry and the public sector to create innovation. So I'm going to talk about aquaculture today, but I want to elaborate on something that we call the fertilizer crisis. And as you know, we are totally dependent today on artificial fertilizers or mineral fertilizers to produce enough food through agriculture to support the world's population. And what we call the fertilizer crisis refers to the fact that this dependency on artificial fertilizers is not sustainable. So first of all, the production of nitrogen fertilizer consumes 2% of the world's energy, which is a lot. And we also produce phosphorus by mining phosphorus rock, which is a limited resource that we're going to run out of at some point. And then there's the other problems like ammonia release and runoff and eutrophication and soil health and desertification, electricity costs and supply chain issues, like super relevant right now. So it's a very complex issue, and we need sustainable alternatives to these mineral fertilizers. And the reason I mention this is, of course, that aquaculture releases vast quantities of nutrients in the form of sludge and in the form of dissolved inorganic nutrients into the environment. And this is problematic because it contributes to eutrophication of water bodies, and it's also a waste of resources that we should be taking advantage of. And this is, of course, not a new issue, and it applies to many industries, and we're beginning to see regulations that target these nutrient release from industry, like the Industrial Emissions Directive from EU, which entered into force in 2024. It's not yet targeting Norwegian aquaculture, but probably they will be targeted sometime in the future, so that they need to reduce the nutrient release to the environment. So anyway, if you look at these numbers, the total release of inorganic nutrients from mariculture in Norway, and you compare them to the volumes of inorganic fertilizers we use in Norway for agriculture, we don't have a lot of agriculture, but you can notice that we could, in theory, get most of all of our fertilizer requirement from aquaculture. And in practice, of course, that is difficult to achieve because Norwegian aquaculture is mainly done in open net pen salmon farms, where it becomes very challenging to recover inorganic nutrients. But this limitation is beginning to shift with the rise of land-based aquaculture in RAS systems. So in Norway right now, there's probably about 100 land-based facilities. Not all of them are recirculating aquaculture systems, but all of the new ones are. And these modern recirculating systems display two key traits that, if you compare them to sort of conventional aquaculture, greatly reduce the threshold for recovering plant nutrients. So first of all, these modern facilities are fully dependent on water treatment systems to recycle water. So when this infrastructure is already in place, the economical threshold for adding additional technology to start recovering plant nutrients is reduced. And also commercial systems today, so the land-based facilities, they operate in fresh or slightly brackish water, so not seawater. And that enables us to produce organic fertilizers suitable for plant production systems in which sodium and chloride might otherwise represent a problem. So the product I'm going to talk about today, which is called Nutricycle, is developing technology to recover dissolved inorganic nutrients from effluent water. The project is an innovation project for the industrial sector funded by the Norwegian Research Council. We were granted approximately 19 million kroners, which is the equivalent of 2 million euros, approximately. 3 years duration. We actually finished the project over Christmas. And it's owned by a private company called Colombi Farms. And they have produced this video in the project that sort of shows the physical setup that we've installed. So this is from the RAS facility that we've built the system in. It takes the effluent water, treats it, creates two water streams, one of which is clean water. You can see it to the right, which goes back to the fish. And the other stream is the inorganic concentrate. And we use that in our vertical farm to produce leafy greens to test it as a fertilizer. And then we've also performed trials in sort of traditional hydroponics, open hydroponic systems, and also in open field cultivation to test it as a substitute for mineral fertilizer in traditional agriculture. I'm just going to let the video finish. So I can't share the details about the technology because it's owned by this company, but they have allowed me to say that it's based on ultrafiltration and reverse osmosis. So we basically built this sort of water treatment facility inside of next to the recirculating aquaculture system that uses ultrafiltration and reverse osmosis to create this in nitrogen-rich organic fertilizer based on the Raus effluent water. And then we also produce clean water, which goes back to the fish. And in the project, we designed 2 studies to test our concentrate. And the first one looked at the storage stability of our concentrate. So one of the key features of conventional fertilizers is that they are stable for a very long time. So you don't have to worry about nutrient integrity or microbial degradation during long-term storage. And that's obviously very important because it means that consumers like farmers can stock up with everything they're going to need for a full growth season or even multiple seasons without worrying about quality loss. So we realized pretty quickly that if we're going to have a chance at establishing our concentrate in any market dominated by conventional fertilizers, we would need to ensure that our product also displays long-term storage stability. So we designed a study where we stored our concentrate for 6 months with and without pasteurization. And then we took samples at regular intervals and had them analyzed for chemical and microbiological stability. And the results following 6 months storage shows that our concentrate is both chemically and microbiologically stable during storage for at least 6 months. And we actually just resubmitted the manuscript today. It's a minor review, so we hope that it will be published soon. And the conclusion based on this study is that our nutrient concentrate that we produce from effluent water is suitable for long-term storage as a nitrogen-rich organic fertilizer. And we believe that the ultrafiltration plays an important part in that because it removes most of the microorganisms before storage. All right, and the second study, the background was to simply test if we can replace conventional fertilizers with the concentrate that we produce from the effluent water and to what extent we can replace mineral fertilizers. So we designed this study, cultivated 4 cultivars of leafy greens in our vertical growth house. And one of the challenges is that it's not possible to fully replace mineral fertilizers using this effluent concentrate because there's some compositional imbalances so that you can't really get a balanced plant fertilizer out of the effluent water. For example, in today's aquaculture, at least here in Norway, they add components to the feed, which means that the phosphorus is deposited mainly in the sludge rather than the dissolved fraction. So we have to balance our inorganic concentrate with mineral fertilizers to get optimum plant growth. But we were able to replace a total of 65% of the mineral fertilizer and 90% of potassium and 70% of nitrogen with our concentrate without compromising the yield or the product quality in our hydroponic setup. So the conclusion is that we can replace, or at least we can alleviate the need for conventional fertilizers in hydroponic plant cultivation. And the last sentence there, ongoing trials to test open field cultivation. That's what I'm going to end my presentation on, because this is what we call it the challenge of volume. Some people call it the opportunity of volume. But anyway, I want to exemplify it with this facility, which is owned by Hema Seafood. It opened in 2024. It is a pretty impressive facility. It contains 16 individual RAS facilities, and I think it cost more than 400 million euros to build it. And it's scaled to produce 8,000 tons of trout per year. And 8,000 tons is not really a lot when you compare it to Norway's total production, which is 1.5 million tons of salmonid fish. But this facility alone will produce more than 700 tons of nitrogen as waste per year, if we make some general assumptions about the sort of the feed conversion ratio and the nitrogen content of the feed and the assimilation in the fish. So anyway, based on a nitrogen demand of approximately 2.4 kilograms of nitrogen per metric ton of tomatoes, this facility alone generates enough nitrogen to produce approximately 290,000 tons of tomatoes, which is 8 times Norway's total consumption per year in 2022. So the point I'm trying to make here is that the solutions we develop to valorize inorganic nutrients from aquaculture need to be scaled to these large quantities of inorganic nutrients. So we can't just sort of install a greenhouse at the end of this facility and supply leafy greens to the local population. That's not going to off-put a significant portion of this inorganic component. So to continue this development, we want to get funding to see how we can integrate this technology that we've developed with conventional agriculture, because conventional agriculture is the only sector, at least here in Norway, that is scaled to off-put significant amounts of these inorganic components. And I guess one of the challenges is that they never built these facilities in order to recover inorganic nutrients. They're just scaled to the local water body. They get a permit based on how much water is running through that river next to the facility. So with that, I would just like to say thank you for the invitation to speak, and feel free to reach out if you have ideas for collaboration or ideas for how we can sort of move forward with this concept that we've designed in this project. So
Thank you so much, Jon. And it's actually a very interesting point that you just left to us and to the audience is the dimension and the scale that we need to actually address the problem. So thanks also for your thoughts on that. While you were presenting, you know, one question came to my mind. You were concentrating in RAS effluents for plant fertilizers. And I was considering what microbiome management strategies do you recommend to prevent pathogens carryover while at the same time you preserve some of the nutrients that you just mentioned. So basically, how do you ensure that you have something that is not contaminated? What do you do with the pathogens?
Right. Yeah, so we worked closely with expert in hydroponic cultivation, and my understanding of like the current hydroponic industry is that they maintain rigid control of the growth environment. So they don't allow sort of, they try to minimize the exposure of pathogens into the system. So we used ultrafiltration to make sure that the concentrate that we produced was free of any sort of carryover microorganisms from the fish system. And sort of, I guess one of the challenges as well when we're talking about hydroponic systems is that my understanding is that they prefer to use mineral fertilizer because it allows them to maintain this strict control of the growth environment. So our solution was just to ensure that they didn't enter the system, but we had hopes and we had ambitions to try and include that portion of microorganisms that we sort of removed from the fish system. We wanted to include it to our fertilizer to see if it could have a probiotic effect on the system. But we never got to try that. And I'm not sure if our hydroponic person really were keen on that idea, because it would mean a lot of cleaning and maintaining the system in the follow-up months. Did that answer your question, Claudia?
Yes, that's interesting. So what we are seeing is that we are at a fairly late early stage of development. So let's finish one stage before we can increase the complexity of the other stage. Yeah, that was interesting. Thank you. We have another question. So in an open water, so net system, isn't RAS system an everyday challenge due to increasing water pollution? So it's now let's turn to the environment and let's see how pollution is everywhere. How is this affecting RAS systems? Is this a challenge for you?
If I understand this question correctly, you're asking about the water pollution from RAS systems and open water systems?
Yes, open water systems. Yeah,
Okay.
I understood from the open water systems mainly.
Okay. So in our open water systems, we have, I mean, Norway has a big advantage because we have a very long coastline. I think we have the second longest coastline in the world, and we have access to this massive water body that we can release everything we want into. That's been the sort of the general consensus until now. And then with the new emissions directive from EU, at least the land-based industry, they're sort of scrambling right now to find solutions. And the only thing they know is that it's going to be very expensive to start treating the water that they have been releasing so far to sort of remove some of these contaminants. So we're in a transition right now, and I think RAS systems will be, when we're talking about aquaculture, they will be the first ones to be targeted by these new regulations. I think Norway sort of trying to maintain the relationship with EU, so they've sort of inserted these new regulations also in Norway, even though we're not part of the EU officially, but we're also given leave to the most important, the key industries, to continue their operations. But I know that this facility that I presented, the HEMA seafood system, I think they spent 300 million kroners just on their water treatment system to treat the water coming out of the system. So it's one of the main barriers for why RAS still hasn't sort of taken off in terms of commercial value yet. They still have trouble competing with these open net systems because Norway just allows them to continue as usual without worrying about the output.
Yeah, that's interesting. Well, that's a good question and a very interesting topic. Thank you for raising that question from the audience. Just one final question, Jon. You mentioned that you have conducted studies about the duration or the shelf life of the biofertilizer, but have you addressed the question of delivery mechanisms? So encapsulation, and so how is it going to be used by the industry?
Yeah. So we did perform like feasibility analysis of actually integrating this for conventional agriculture, and the first challenge you run into is transportation costs because it's mainly water, right? I think our highest concentrate had about 7% nitrogen, so it's still mainly water that we're trying to transport around. And in Norway, I guess the main challenge is that we don't have a lot of agriculture, and it's sort of spread out in a big area. So I think our sort of best idea to solve that is to go to countries where you have high density of agriculture, and if you have a RAS facility or you're planning to make one close to that area, then you can try to include drip water irrigation systems so that you can transport that water directly out into the field to provide irrigation and fertilization at the same time. But yes, I guess the main challenge of our concentrate is you're going to have to either concentrate it even further, maybe make a granulated product that you can transport, or produce it close to where it's supposed to be used.
Okay, thank you so much. I think these are all very important aspects to make an industry work, but at least you have them on your mind. So also congratulations on the project and best of luck. Let's try to keep in touch to see whether we can have synergies and join forces to move forward this type of solutions. We all need it. Jon, I'm going to move on, but you have a question in the chat. So could you very kindly address the chat while I will call Etiele Morais. Apologies. Maybe you can then explain your background because this is a very Portuguese name. Etiele, a researcher from UPC, Ramon and Cajal. It's a pleasure to have you here. Etiele, when you're ready, please, the floor is yours. You can start sharing your screen. Thank you.
Okay, I'm gonna share my screen. Are you seeing?
Yes, it's perfect in presentation mode. Thank you so much.
Okay. So, good afternoon, everyone. I'm Etiele Morais, so I'm Brazilian. That's why I have this similar to Portuguese name, surname at least. As Claudia said, I'm a researcher at the UPC. And I'm going to talk with you today about the wastewater for sustainable biofertilizer and biostimulant production from microalgae. So I'm going to start first telling you about who are we. I make part in the GEMA, the Group of Environmental Engineering and Microbiology from UPC, and we are based in Barcelona. Our research topics are main focus on developing sustainable solutions within the circular bioeconomy, mainly on wastewater treatment. We have a strong expertise in wastewater treatment and resource recovery, not just with microalgae, but also by other technologies, and using these streams as alternative and sustainable feedstocks. In the case of the microalgae, is one of the topics that we work, and we focus mostly on the application of the biomass for the obtention of high-value products, including biofertilizers and biostimulants. In this topic in particular, we have been actively involved in some national and European projects focused on that in the last years, where we explore this potential of the microalgae-derived compounds to improve the plant performance and resilience. So, to start a little bit different from my colleagues, they talked mostly about the problems that we're going to solve with the biostimulants and the biofertilizers. I want to explain first the main difference between these two. So, biofertilizers mainly provide directly nutrition to the plant. So, as microalgae are rich in nutrients like nitrogen, phosphorus, and potassium, as well as micronutrients, their main effect is to support plant growth and increase yield through nutrient supply. On the other hand, the biostimulants will work different. Instead of supplying nutrients, they stimulate the plant's physiological process. These products are usually based on microalgae extracts, different from the fertilizers that directly is the biomass. And these extracts are rich in bioactive compounds such as phytohormones, amino acids, antioxidants, and extracellular polymeric substances that can help to promote stronger root development, improve stress tolerance in plants, and also better nutrient uptake. So, what about it then to produce these products in wastewater? There are several advantages, as microalgae can remove nutrients from wastewater, promoting the resource recovery. And this can increase mainly the sustainability of the process of wastewater treatment compared to the traditional wastewater treatment systems. So we can also observe a cost reduction mainly on the products of retention by microalgae using the systems. Mainly when we focus the work on biorefinery, when you have more than one product that can be obtained from the same biomass, and also when you have the focus on circularity. And about circularity, an advantage of microalgae application is that capacity of closing loops in industrial systems. In case of biostimulants and fertilizers, for example, we can treat an effluent from an agro-food or a food industry, and the produced biomass that treat these effluents can be used for biostimulants and biofertilizers that can in the future be applied for the cultivation of these crops that's going to be used again for the obtention of the industrial products. So we start our journey on this plant enhancers production with the fertilizers, asking ourselves if this microalgae grown in the wastewater are able to be applied as fertilizers and also reduce the application of the inorganic fertilizers. So in this study specifically, we evaluated the performance of microalgae biomass culture in domestic effluents applied to basil plants and compare it with conventional fertilization. The results show that the plant growth was comparable across treatments. We also did a treatment mixing microalgae fertilizers with the inorganic fertilizers. And in some cases, even improved the growth when microalgae were used. However, microalgae alone could not fully replace the inorganic fertilizers in terms of nutrient supplies mainly. And the key conclusion of this study was that microalgae could effectively replace up to 50% of inorganic fertilizers, maintaining the plant performance while reducing these chemical inputs. But we kept asking ourselves if the wastewater composition could be a problem for the fertilizer production because of the possible presence of pathogens and heavy metals, organic pollutants. And we asked ourselves was that, can microalgae remove these compounds from wastewater? Did they stay on the biomass? Could they pass to the plants? What happened? And searching, we saw that it depends on wastewater source, on microalgae strain, if this microalgae strain, which kind of removal mechanisms they have, and also the plant where we apply it. So we decided to work with what we had, and in another study of our team, we also looked for the presence of contaminants, either in the biomass, but also if after the treatments, they appear in the plants cultured with the microalgae biofertilizers. So we kept using the microalgae produced in domestic wastewater, and now for application in lettuce. And the results show that although wastewater was used to grow microalgae, contaminants from emerging concern and pathogens were removed and mostly didn't pass to the microalgae biomass. And more importantly, these substances, when we apply the microalgae to the plants, were not detected in the final agriculture product, that was the lettuce, and that indicates that the use of microalgae biomass could be safe and do not show risks for applying in food. Then we also ask ourselves, what about the cost of the fertilizers, the sustainability? Is this real sustainable? And then we have this other study where 3 different scenarios for lettuce cultivation were compared to evaluate environmental and economic outcomes. So we consider the growth of lettuce in the condition of the first article that we published with microalgae fertilizers only, the inorganic fertilizers, and the combination between these two. All scenarios include the full life cycle of fertilizer production, transportation, and application. The environmental impacts were assessed too, and the microalgae only scenario performed best environmentally in 10 out of 11 impact categories that we studied. Economically, also, we have good results. So the results there suggest that microalgae grown wastewater can be 30% cheaper compared to the other scenarios when we talk about culturing the lettuce. And after this, we, not after, but also at the same time, we started to explore also the biostimulants, and we started studying in the biostimulants. So we first have the evaluation of the capacity of production of pheromones by different microalgae strains culture in the wastewater. And we have a pretty promising pheromones composition, some for germination objectives, other for the stress, other for growth of microalgae. And then we select one of the strains to continue our studies, mainly based on the capacity of treat the wastewater too, because we are mostly focusing on the wastewater treatment. And then in this study, where we cultured the Senendesmus microalgae, was the one we selected. We studied different kinds of extraction, the physical and chemical, and applying it on lettuce, we notice an increase in fresh weight and also increase in chlorophyll content in plants. So improving the development of these plants with the biostimulant produced. So now, aiming to continue this, we are looking for applying microalgae as biostimulant for improved plant resilience to stress, mainly salt and water stress. We already have some promising previous results, like different pheromones profiles of microalgae culture in different effluents really make difference for using different applications like germination of just improvement of the resistance and resilience of the plants. And that we find it interesting because that can be a key for the formulation of new biostimulants based in microalgae in the future. So as conclusions of all this, like a takeaway message, every year wastewater carries nutrients, contaminants, and pathogens that threaten our soils and crops, but microalgae can turn this problem into a solution. And they grow, the microalgae grow in wastewater, remove these harmful substances, and can produce these biofertilizers and biostimulants that can be safe to the crops. Applied to the plants, they can enhance the growth, and this approach mainly closes the nutrient loop, reducing the chemical fertilizers that are one of the main topics, and moves us to the circular, sustainable agriculture systems, also wastewater treatment systems. And I would also like to leave some main challenges that I can see from working not for a long time with the biofertilizers and also biostimulants for microalgae, but it's really complicated still, even if we see that the costs are reduced, to develop this more cost-effective methods for harvesting and processing microalgae biomass. This could improve a lot the commercial viability of these biostimulants, these byproducts from microalgae. Also, ensure that these microalgae biofertilizers and biostimulants meet agricultural and environmental regulations can be challenging, and also to meet these environmental regulations, maintaining a consistency of the nutrient content of microalgae biomass in wastewater is complicated because the wastewater composition change day by day depending on where the source you are applying. So that was it. That's my contact. We can also find us in LinkedIn. And thank you so much for the invitation to participate in the webinar.
It's a pleasure. It was great to have you here, Etiele. Again, very good food for thought that you leave us here, particularly the challenges that we need to be looking at when we consider this business. So thank you so much for a great presentation. We have a question. Marina would like you to comment a little bit more on how microalgae provide macronutrients to plants, of course, as biofertilizers. So what is the process? Can you explain a little bit more about this process between the microalgae and nutrient supply to plants, macronutrients?
Microalgae, when applied as biofertilizers, we most apply it by the biomass directly. The biomass are mostly rich on these compounds, the phosphorus, the nitrogen, the phosphorus, this can, we saw that in some studies, not ours, but from other references, that one of the main advantages of applying microalgae biofertilizers are the slow release of these nutrients to the soil, mainly because of the structure of the microalgae cells. But mostly the application are directly mixing the biomass with the soil.
Okay, thank you.
Answer your question.
Yes, it was very clear from my side, but these are, fairly, I can see a thumbs up from Marina. Thank you. Etiele, I had another question for you. I don't think you have addressed it in your presentation, but you were mentioning about microalgae-based biostimulants from wastewaters. Which strains do you think show the best field performance for, some of the functions that you've described, namely for salinity soils or the general soil health regeneration? So which strains are we talking about?
So actually working with wastewater, we also first look after the microalgae strains that could perform best on the wastewater we are treating. Because even if we have a microalgae with a good potential for specific phytohormone production, that could be, for example, for the stress conditions and everything, it's important to have a significant amount of biomass, even if biostimulants don't demand this amount of biomass, like the biofertilizers, for example. But we always try to culture these microalgae that are already adapted to the wastewater that we're working with. And after that, what we try to do is to evaluate this phytohormones composition and also which other biocompounds that can be produced by the microalgae. So if you have a profile, for example, Synetesmus, we produce have a phytohormone profile that it's suitable for the stress conditions that can regenerate and help plants to be resilient to mostly water. That was the first one that we are trying now. But for example, there are some microalgae that can produce EPS, isopolysaccharides, and isopolysaccharides are really known for being applied as also a good regenerator of the soil in case of drought. So drought conditions. So it should be evaluated. Like in our case, we always look first for the microalgae that perform better in the wastewater that we are trying to apply.
Okay, that's an important starting point. Although from a farmer perspective, I think the main goal is that they achieve, a certain function. But I fully understand that we need to start somewhere, and for you, it's actually easier, and we can then use those results. Thank you so much, Etienne. It's just, yeah, I will ask you to stay a little bit longer for you, George and John. But in the meanwhile, let me just tell our participants that while you're here, we invite you to do, we call it the follow assessment, okay? It's not really an assessment. It's just for you also to test your knowledge and to see if you have learned something throughout this webinar. It's you already also have the link here in the chat. Thank you so much to the team. They are great. They are always doing everything with a perfect timing, but also with the QR code here. So we invite you, and even the speakers can also have a go to see if you get it all right. I'm sure you will. These are very friendly questions. So this is the way that we are going to use to issue the certificates, and we are very happy to wish them. It's just for you to have to fill in this very short questionnaire. And just before we go, I think we have very interesting approach. I know at some point we were saying that we were covering the same topic. I don't think it's any problem. It's not ever enough to go through the same issues again and again, because they are actually very important. They are a bit cutting edge in terms of the type of solutions that we need. We had several approaches: real-time microbiome monitoring, RAS effluents, what I would like to hear from you and from the perspective that you take in each of these areas. How do you think these areas could be integrated in the future in a biotech platform such as the Ampliaqua? So think about the synergies that we can have for real-time monitoring fish effluents, microalgae as biofertilizers, and to ensure that we have a truly circular food system. I would like to hear your views on this. Oh, no one? So I was heading to you. So it's how do you feel that the what you have been presenting here, it's complementary? Where do you think are the main challenge between this John, you mentioned some of the challenges in terms of scaling out and the open air water. You also touched about wastewater variability at the end. So what we need to have, a full round system that integrates everything that you mentioned here.
And I can provide some input from our experience. So I think the main challenge we have when we're working with something like effluent from aquaculture is that we're competing with a low-cost product, and we're designing a technology that's relatively expensive to operate. So there's a lot of development still ahead of us just to sort of establish an economically feasible way to compete with conventional fertilizers, because they are still so accessible and relatively cheap. So there's really no big incentive for the consumers to switch to some alternative. I mean, it just means more unpredictability for them. They want to know what they're buying, and they're sort of used to getting mineral fertilizers. So it's a technology that needs to be developed. It's reducing costs in all levels of the value chain, and also convincing customers that they can safely buy something that's not a conventional fertilizer, and then start integrating these additional technologies, for example, microalgae cultivation. But it's so hard to compete with the low-cost products with these high-tech solutions.
John, interesting thoughts. One question on that. Do you think that this is really an obstacle, or do you think this is just part of the technology development stages, that when we start, of course, it's going to be expensive?
Yeah. So if I compare it to, it's some overlap to what we see in aquaculture in Norway. So, sorry? Oh.
We need to bring the industry to find ways. Sorry, I don't know if you got my question. My question was, if you see that this price burden or the price of the technology, or do you think it's just part of the process of any technology development? So it always starts, as being quite expensive, not available to everyone, but as we develop, as we democratize access to everyone, we are expecting these technologies to become cheaper and cheaper.
Yeah, I think both. So part of it is developing the technology, and then what we see from other industries, like in Norway right now, the reason that land-based aquaculture is gaining so much traction is because sea-based aquaculture is becoming very expensive because of all of these welfare issues that you get when you increase the density of your production. So suddenly sea-based aquaculture is becoming so expensive that land-based aquaculture can actually compete with that. And we see the same now with cod aquaculture. That's also beginning to get established because prices of cod are so high compared to what they were the last time they tried, which is some years ago. So once you have that,
you tell that to a nation like Portugal. You tell that to a
right?
nation like Portugal. We are
Exactly.
huge consumers of cod.
Yeah,
Thanks for bringing
exactly.
that up.
But I think once you get to some level where, for example, fertilizer becomes very expensive, and you start to see that these new systems can actually have commercial value, then you will get a rapid development of technology and sort of wide synergy with the agriculture and industry, hopefully.
Hopefully, thank you. Good points. Also, at least she also adds that regulation is very strict for biomass coming from wastewater, and this can also adds costs, of course, and it's difficult and expensive to prove claims under the current regulation. So policy also here working as a barrier, but hopefully as draw more attention to these issues, we can actually start to make some pressure for policy change. Jorge, Etiele, would you like to make final comments?
Yeah, no,
Yeah.
I will just add, just like the comment of Alice, that I think that more than the technology, that was something that I also talk about, the harvesting and from microalgae biomass, harvesting and other culture conditions that could be improved for reducing costs. Also, we have this difficulty on meet the regulamentations, because microbiology is not explicitly cited in the regulations, and also we need to fit in another categories that is not completely exactly what microbiology means, and mostly products that have microalgae should be mixed to fit on the regulation. So it's also complicated, as Alice said.
Thank you for your views. And George, we had the price of the technology.
I
What's your
lost you a
view?
bit there. You're asking about the price of technology, right? Claudia?
Well, we have talked about the price of the
Yep.
technology. What is your view, George?
So specifically regarding our project, so yeah, in the beginning, those types of monitoring systems involving real-time technology are a bit capital intensive, but it also comes of economy of scale. If someone invests in that kind of sensors, they will be cheaper with time, and if they have a higher production. But mostly, I believe the informatics itself is relatively accessible, relatively easy to use. In terms of cost of operation, and Jan already addressed this issue a bit. Yes, gas production, indeed, it's more expensive. It's more capital intensive and more difficult to implement in the beginning. But there are also another possible return of that investment. For instance, if the consumer trends are looking for high quality products, and by that I mean like high quality fish, high quality salmon. Since we have closed systems that we do monitor everything that is going on regarding the environment of fish, if you can guarantee the health of the fish and the quality of the water, we can make the product like certified and make the consumers know that this is really high quality. To grow in this grow in these conditions, and in like with they can track also with certificate tag how that high quality fish was produced, and the fish can also be used eventually for high quality ingredients. So that's all different market there. So basically, what I'm saying for the near future, like that, high high demanding, but also a segment of the population where have a possibility to pay for better products could cover some of the expenses. Eventually, some of those technologies can become even more cheaper. That is something that is seen for many technologies, namely microbiome, NGS, which analysis, so to evaluate the microbiome of the samples are becoming twofold or threefold cheaper each 5 years. So it's possible that those monitoring technologies will be really accessible in the future, and will also be easy to implement, and hopefully the REST systems and also those negative consequences Jon was addressing in terms of how are you going to take care of taking care of the wastewater and effluents will also we can also have some ways of dealing with that easier.
Okay, thank you, George. Thank you also for ending up on
We can hear you, Claudia.
really sorry. So it's just network
We cannot hear you.
work.
Sorry, Claudia.
I will close the webinar. Cannot hear me?
Yes, now it's okay.
Yes. Okay. So
Yes.
I'm really sorry. I was just saying that, George, thank you so much for ending in a positive tone. It's just sustainability and circularity and food quality are really for what we see is not willing to pay this premium price. Let's see if things change in the future so that we can kick off this system with at a high. This webinar has come to an end. I do apologize to everyone for my internet connection. So sorry things happened, but I hope you have enjoyed. I think we had great discussions, great views, addressing very important topics, the potential and the challenges of the systems. Also, it's good to see that you want to see more of this disseminated and communicated. That's why we are here. As you know, we will continue with these sessions next week, and we also have the open days for visits at Ampli Aqua, the Ampli Centro. You can visit us on our website, register for the open days, and we'll be very pleased to receive you and to welcome you at our facilities to explain you a little bit more about the system. Okay, that's all from my hands. Also, a big thank you to the speakers, to the team, and of course to you for being always here. So I hope to see you again next week. It was a pleasure. Bye.